everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by our guest, Dr. Lena Ekström. So, Lena, we are so excited to have you with us today. How are you?
Yeah, I'm fine. Thank you. And thank you for inviting me.
Well, we're excited to have you with us, and we're excited to learn more about you and your work. But to give our listeners a little bit of background, Lena is a hospital chemist at Karolinska University Hospital and an adjunct professor of pharmacology in the Department of Laboratory Medicine at the Karolinska Institute, where she specializes in doping research. So today, our conversation is going to focus more on her work, examining a variety of different factors that can influence anti-doping test results, including interactions with other substances, genetics, hormone cycles, biological variability, and microdosing. So I'm excited to get into all of these details, but perhaps first, Lena, can you tell us more about yourself and your roles at the Karolinska University Hospital and Karolinska Institute?
As you said, I have joint positions, both at the Karolinska Hospital and at the Karolinska Institute. So my major employee is the Karolinska University Hospital, and here I am employed as a hospital chemist, as you said. And the doping control laboratory in Stockholm is situated here at the Karolinska Hospital. So it's here where I mostly spend my time working with routine analysis and also as a research manager. And I'm also partly employed at the Karolinska Institute, having a professorship. And in addition to doing anti-doping research and supervise PhD students, I'm also involved in teaching, meaning that I also supervise undergraduate students in different bachelor and master programs here at KI. And my teaching area is in clinical pharmacology, and I have courses and lectures mostly at the biomedical science program. So I would say that I'm in a very good position where I can combine my academic work with clinical practice. So having this kind of diverse workload, it never gets boring.
Definitely. I think having that variety day to day can be really fun. But I'm curious, where did this interest in science and perhaps anti-doping come into play? And how did you find your way to your current positions there, Lena?
In my case, I was in the beginning not at all interested to work in the academic world. I never planned to do PhD, less so continue to do research after that. It was more that things just happened. I was offered a PhD position after I finished my master project in biochemistry. And then I got a PhD in toxicology here at Karolinska Institute. And then I got the opportunity to do a postdoc in San Diego. And I went home after 1 year in California. And then I was looking for a real job. But then I came across an ad where they looked for a postdoc in an anti-doping research here at Karolinska. And this was in 2001. And the professor here at the department, Anders Rane, together with the doping control laboratory director, Mats Skale, they had received a big grant from WADA for this postdoc project. So I started as a postdoc and got involved in anti-doping research. But at that time, I also did the research in other areas, like in pharmacogenetics and some cancer research also that was going on here. And after 4 years postdoc period, I was hired as a lecturer. And I got more and more teaching obligations. I mean, I really enjoy teaching. It's fun to interact with students. And also by teaching, I think you expand your own knowledge. So it's a very stimulating environment. So I like the teaching. And I also, during these years as a full-time lecturer, I also continue to do research. And then it was the anti-doping research that I find was most fun and interesting. So I focused more on that research. But over the years, it was hard to maintain high quality teaching and research because a lot of administration duties and so on. So I quit my position at the Karolinska Institute in 2018. And then 1 year later, I got this offer to be employed by the Karolinska Hospital instead. And like now having this joint position.
Oh, very interesting. So what was it about anti-doping research that really captivated you or made you think it was really interesting and fun?
I think the environment, like the anti-doping community, and it was more a friendly environment than perhaps in cancer research and some drug research, because of course you compete when you do research in anti-doping as well, but it's a more friendly way.
Absolutely. I think having this relatively smaller field where it really feels like a community can be really rewarding.
We work for the same purpose. We want the same things.
Exactly. Well, let's talk about the research that you do. Can you maybe give us an overview of some of the topics and areas that you conduct research in your lab?
Roughly, I can say that there are like two types of anti-doping research projects that I am doing. And the first is detection of doping substances. And perhaps the most exciting research is to perform administration studies to see if you can detect doping substances that you give to like healthy volunteers. And over the years, I have done administration studies with mainly testosterone, but also other steroids and growth hormone and EPO. And lately focusing more on microdoping and detection of low doses of hormonal drugs. An ongoing study now is that we have given volunteers a single dose of both testosterone and recombinant EPO to see if there is an interaction between them and if they affect the possibility to be detected. And administration of microdosing is research that we are planning to continue to do. For example, in pipeline, we have a study to administer recombinant EPO to women and also administer testosterone to women. And I also, over the years, I have done studies to study the natural variation of endogenous markers that are included in the Outbreak Biological Passport. And so I have done many studies how different drugs, exercise, pregnancy, menstrual cycle, etc. affect urinary steroids in the passport. And now lately we have also switched focus to conducting research. For example, drugs training affect the serum steroids, testosterone and androstenedione, which are the serum steroids included in the serum steroid module of the passport. And the other anti-doping research area that I have an interest in and have conducted over the years, it's to learn more about the use of anabolic androgenic steroids outside of sports. I mean, the use among recreational athletes, gym goers, but also people not involved in any sport activity at all. And here at the Karolinska Hospital, we have in our department the Drug of Abuse Laboratory, and this is a sister lab to our doping control lab. So here we have over 2,000 samples per year that are analyzed for anabolic androgenic steroids. And many of the patients are positive on the steroids. So these urine samples, they come from patients that are in drug treatment programs, substance dependence care, or samples from workplace testing. So the research I do with these samples is to reanalyze them to see what kind of other doping performance enhancing drugs are used or abused together with the steroids, such as selective androgen receptor modulators, for example, but also how common is that people that are using steroids also abuse narcotics. And in addition to analyzing these hospital samples, I collaborate with an endocrinologist that meet patients regularly that declare use of doping substances and from where we also can get samples. So we also have an anti-doping hotline here at our department, which I worked a lot together with over the years to create people that are declaring that they're using steroids.
Well, I think this is remarkable. And I know, Lena, you've been working on a lot of really interesting projects over the years. And I know back in 2016, you were actually awarded funding from the Partnership for Clean Competition to examine potential interactions between testosterone and the drug codeine, a common medication that's used to treat pain or cough. So let's talk about this. I think this area of combinations of different substances is absolutely fascinating. Can you talk a little bit more about the background for this particular project?
Yes. I was awarded from PCC something called the microgrant. I must say, I really think the microgrants is an attractive approach to financially support smaller projects that could be done within a short period of time. And the reason we did this particular study was that we had a hypothesis that the use of codeine could influence the chances to detect testosterone doping. And as you said, codeine is a quite common analgesic medicine that is not prohibited. And testosterone is one of the most popular steroids. So I think it's a clinical relevant interaction to study. And the most important marker to detect testosterone doping is to measure the ratio of urinary concentration of testosterone and epitestosterone, the T ratio. And both testosterone and epitestosterone are metabolized with similar phase 2 enzymes as codeine. So there could be a potential metabolic interaction between them, which we wanted to investigate in this study.
Very interesting. And then what were your findings of the study? And perhaps you could also share why it is so important to examine these potential interactions between these relatively common medications and measures that are being used in anti-doping tests.
Well, for this study, we could conclude that there was no interaction of direct clinical relevance. I mean, the chances to detect testosterone using the urinary T ratio as a marker was the same as when we administer testosterone alone. So maybe this is a bit boring when you do research. I mean, I think it's more exciting when you find an interaction that proves your hypothesis. But at the end of the day, also these so-called negative results are important to publish in order to get the whole picture of something. And particularly so in anti-doping to understand which drugs do not interact with these doping markers and which drugs may be considered confounding factors. And such interaction knowledge can be of importance when interpreting the test results. However, we noted in this study that only after 3 days intake of codeine, the endogenous serum testosterone levels decreased significantly. And in some of the participants, the decrease was very evident. I think more than 50% drop in serum testosterone concentration.
Oh, wow.
It's well known that the use or abuse of high doses of opioids can result in a decrease in testosterone production, but that codeine decreased testosterone levels already after 3 days was a bit surprising. But at that time, serum steroids was not of interest in doping testing, as only urine steroids were analyzed back then. But now, as we have implemented the serum steroid module of the passport, I think it would be interesting to perform more studies how codeine and also other painkillers might affect the disposition of serum steroids in the passport, and perhaps also include more steroid serum markers, and of course also include women in the future study, because in this particular study it was only men that was included.
Certainly. And I think that's a pattern I've seen in a number of anti-doping studies where a lot of the research has been done in men. So there are still many unanswered questions in women, particularly related to these hormone measures. And I think genetics are another thing to kind of keep in mind when we're talking about confounding factors. And there's some genes in particular that I'd like to touch on today. So can you explain how your work on some of these factors like genetics can impact doping tests for testosterone or other anabolic androgenic steroids?
Yes, pharmacogenetic is an area I have been working a lot with over the years, and I am in general very interested to investigate how genetic variation might impact different kinds of phenotypes, including, for example, doping biomarkers. And there is particularly one genetic variation. It's the UGT2B17 gene deletion that we discovered a long time ago during my postdoc period, and showing that people that are homozygous for this UGT2B17 gene deletion, they excrete low amounts of testosterone in their urine. So they subsequently have a low T ratio. And they also don't have the capacity to excrete exogenous testosterone. And hence, they can escape traditional doping tests for testosterone. And I think this was actually one of my most exciting research findings so far in my career. But now when we have this passport approach, I think this genetic deletion is not so much of clinical importance, since now each athlete is his or her own control rather than having traditional population cutoffs of the T ratio. But I have also found genetic polymorphism in other genes that might not influence the biomarkers to the same degree, but I still think they are interesting. I find particularly the pharmacokinetics of testosterone. If you give testosterone to healthy volunteers, the serum testosterone concentrations show very large variations. Even though you give the same doses to everyone, they still have very large variation in the serum concentration. And we have identified a single nucleotide polymorphism in an esterase gene that was associated with serum testosterone levels after injection of testosterone esters in men, and another single nucleotide polymorphism in a CLO2 gene that we could associate with an increase in testosterone serum levels in women after transdermal testosterone application. So, yeah, I have many genetics and phenotyping studies.
Well, I think this research is tremendously exciting. And I think you've also done important research, and we sort of touched on this earlier, in men versus women. And I know women have these monthly hormone cycles that could potentially impact the results of their doping tests. So what have been some of your key findings related to looking at steroids and the interactions with monthly hormone cycles in women?
Yeah, so now when we have the passport and longitudinal monitoring of markers in elite athletes, I think it's of interest to study the natural within individual variation of these endogenous doping markers. So in 2018, we conducted a study where we aimed to investigate the within variation during 2 consecutive menstrual cycles in healthy women. And in this study, we found that the fluctuation of the urinary steroids included in the passport, and for particularly epitestosterone, was very large. I think it's already known that epitestosterone fluctuated a lot in women, but here we also show that the reason for this was a strong correlation with the circulatory levels of the female hormone estrogen. And some of the women had very large individual variations, and it seems that the variation was similar between the 2 cycles that we were studying. And these large intra-individual variation may lead to larger thresholds in the passport for the women. I mean, it's essential that the biomarker is somewhat stable if you're going to longitudinal monitoring it. So with that said, epitestosterone appears to be a poor marker in women. And in addition to that, we and also other researchers have shown that T ratio is not a very good marker to detect testosterone doping in women. Of course, you give women lower doses, so you have a not as high increase in testosterone. But it's also so that epitestosterone is not suppressed as it is in men. If you give men testosterone, epitestosterone decreases, but that's not the case in women. So originally, the T marker was developed and studied in men. And as we already discussed before, I think this is a general problem in many research areas that you do more studies in men, and then you just assume that these results are transferable to women. But this so-called shortcoming, I think, has been corrected for in the last years, at least in the anti-doping area, because there have been a lot of research lately conducted in women, which is good. And we have continued to perform studies of the hormone cycle and how it influenced the new serum steroid markers. And we also have done testosterone administration studies to evaluate the usefulness of the new serum steroid markers. And the good news is that the serum steroid markers are more stable than the urine markers in the women, and it's also more sensitive to detect testosterone doping. So hopefully we have a more gender-fair doping test in regard of testosterone detection in place now.
Very interesting. And you mentioned that these steroid markers are more stable in women. Do you still see those patterns of fluctuation over the monthly hormone cycles, or is it pretty consistent?
I think you can still see minor fluctuation, but they are not of such a clinical high relevance.
That makes sense. And I know last year, speaking of within individual variations, I think this athlete biological passport is tremendously valuable. You recently published a paper looking at ABP markers for steroids in urine in males versus females over time. So can you just talk about why this particular study was important to conduct?
This particular study, it was a continuation to study the within variation of the urinary steroid markers, so we can have even more understandable how they vary naturally. And in this particular study, we used data extracted from the ADAMS, Athlete Doping Administration and Management System, and we have test results from athletes that have been tested 5 times or more. And that was over 4,000 test results from over 300 athletes.
Oh, wow.
And the most important result was that we observed that the variation in the urinary steroid profile depended on if the samples was collected in or out of competition. And again, we showed a big gender difference here. Testosterone was much higher in competition compared to out of competition samples in the women. I think it was over 50% higher in competition samples than in out-of-competition samples. But in men, there was no impact of the testosterone levels between in or out-of-competition samples. And this might be due to that much of the testosterone in women are produced from the adrenal glands, which is somehow controlled by stress hormones. and possibly impacted there some stress related going on in competition. So that could have an effect, but this has, to my knowledge, never been truly studied. So that's just a hypothesis. And all these kind of longitudinal studies are important to perform, just as we said, to understand the natural variation that we see in the passport markers. And they are useful for people that are assessing and interpreting the active biological passport.
Definitely. And I think kind of thinking about the big picture, looking at all these studies, it sounds like what you're seeing is there's a fair amount of naturally occurring variation, particularly in these urine samples. So can you share with us how this research is providing valuable data that can kind of inform how people look at the athlete biological passport, and then how that might inform what the next steps are to determine whether this might be a doping case or might not be?
Yes, I think we heard also in previous podcasts here that the passport not so often maybe are used to directly prove that someone has used testosterone or other doping substances, but it's mostly also used if you have suspicious samples, and then you need to confirm it with an RMS test. And with this knowledge, how to interpret the test results, you can decrease the amount of samples that you send for an IMS, because you can see that, oh, this is probably due to contraceptive use, so this is the natural variation that we're expecting based on this study. So you can minimize the amount of samples for confirmation IMS analysis. Of course, that will save both time and money for the anti-doping community.
Absolutely. I think that's tremendously important. And I know you've also recently led a study that was published on detecting microdosing administration. We talked about how this is an important area of your research that I think has been of growing interest in the anti-doping community. And you were looking specifically at microdosing administration of recombinant human erythropoietin, or rhEPO, in dried blood spot samples. So, Lena, can you describe the rationale for this study and then how you conducted it?
Yes, and as I mentioned a little bit, in my research areas over the years have been mostly about the steroids. So entering into EPO doping and EPO detection was kind of new to me. And this particular study was conducted as part of a PhD project of Carmel Highland, who is an EPO method manager here at our lab. And the EPO method, you run the sample on a gel, and then the recombinant EPO you can see as a band or smear on the gel. And this is a direct proof of doping because you don't have this band or smears naturally. So to detect if an athlete has used EPO drugs, it's mostly urine samples that are collected and analyzed, and sometimes also blood samples. So we started some years ago to investigate this possibility to use dried blood spots for EPO detection. And as you know, I think we heard also in previous podcasts here, dried blood spots have been successfully used to detect steroid esters among athletes. And detection of the esters are like recombinant EPO, also direct proof of the use or abuse. So in both these cases, I think dry blood spot is straightforward matrix to use to prove that doping is going on in an athlete. And in this study, we just wanted to explore if dried blood spot could be used to detect microdoses of recombinant EPO with similar rate as traditional urine samples. And prior to this study, we tested and validated the method with spiked dried blood spot in the lab. But at the end of the day, I think authentic positive samples is the best way to really test your method, as we did here.
Absolutely. And I think microdosing is sort of a general term that could include a variety of different parameters in terms of frequency, amount, etc. What were the parameters that you used in this particular study? And do you think they reflect how people might be microdosing in the real world?
Yes, I think there are more studies and anecdotes and stories telling about that the athletes actually use these so-called lower microdosing of both EPO and testosterone. So I think it's very relevant to do studies with microdoping because many of the markers originally was developed when you give very high doses to people. So I think microdosing is something that needs to be investigated. The microdoping dose is different. People say different doses. It's not like Mr. Sharp. What is microdosing? It's a little bit gray zone, I think.
Exactly. I think the definition can vary and the parameters in the real world can definitely vary. So can you talk a little bit about the potential impacts of this work that you're doing in the anti-doping field? I think dried blood spot is something that a lot of people are excited about at the moment.
Yes, I think in this case, the finding that we saw that had a direct impact on our anti-doping work. I think even though this was a very small study, it was only 5 participants, but it was clear that the dry blood spot could detect the administered recombinant EPO to the same degree as the urine samples, or even better in some cases. So in our doping control laboratory, this method has, as I said, been validated and now also implemented as a routine method. And it was recently implemented, so now we are hoping that we will receive dry blood spots from our customers now in the near future. And I think this is an example of research that is closely connected to method development activities that going on in the lab. And I think this is a general strength in anti-doping research, that much of the research that are financially supported and also conducted are clinically relevant and often directly useful and not only nice to know projects.
And I think that's one of the cool things about your position is you sort of span both the anti-doping research side and the methods development side, as well as putting these things into practice. So can you comment on how being able to have a foot in both areas has informed the research that you do and perhaps informed the anti-doping routine work that you do as well?
I think we need to think more about the research that we are doing, that they're applicable very soon. And I think also many of the projects that we are doing is actually with PhD students that are method managers at the lab. So they use their method, that they are experts in a research context. And I think that's also a strength.
Absolutely. And I think research that will ultimately have these real world impacts is really important for the Partnership for Clean Competition. So can you talk a little bit more about your interactions with the PCC, the Partnership for Clean Competition, over time? And perhaps how did you even originally find out about the organization?
I think the first time was when Jenny Schulze, a former PhD student of mine, she applied for and received a postdoc grant from PCC back in 2012 to work in my lab. And this project it was granted was actually to gain more knowledge of hormones and women in anti-doping context. So that PCC grant project was a bit the starting point of my research interest in women and steroids.
That's remarkable. And what have your interactions been like working with the PCC?
Yes, I have the founding that we talked about 2016, and I have entered some of the conferences. It's been really nice. And I think also these digital online conferences been very good to attend. And also the podcasts I have listened to a lot. So I think PCC do a great job.
Oh, thank you. We appreciate your collaboration in advancing anti-doping science. And perhaps you could comment on how you collaborate with other stakeholders, whether it's funding organizations like the PCC or others, and then different labs and other colleagues within this anti-doping space and the work that you do.
In regards of collaboration, I must say my most important collaborations over the years have been and are still with the clinicians. So I have been lucky to work and collaborate in projects with an endocrinologist and also gynecologists, and both of them are very interesting in anti-doping research. And in regards of collaboration with other stakeholders, we have close collaboration with Anti-Doping Sweden, and particularly Jenny Schulze, both in previous and also in future planning studies. And also we do collaborate with the other doping control laboratories, particularly the Paris lab. We share some PhD students together. So over the years, as I said, I have some PCC funding and also have had funding from WADA and also national funding from Swedish Research Council for Sports Science.
Excellent. And I think with the research that you're doing, you're answering some important questions in anti-doping. And the work, as we mentioned, has the potential to have tremendous immediate impacts on the field. But I think oftentimes things are limited, whether it's funding, staff, technology, feasibility of just the projects that you want to do. So if we took away the barriers and we gave you all of the resources you could dream up, Lena, what is one problem in anti-doping that you would most want to solve?
Well, a problem I would address and try to solve is actually a national problem I think we have here in Sweden. There are many people that use performance-enhancing drugs, particularly the anabolic androgenetic steroids, that are not necessarily involved in sports. And as with other illegal drugs in the society, the use can be associated with problems such as adverse side effects. both somatic and psychological problems. And it's not only people using the anabolic androgen steroids that encounter problems, but also family members may be affected, as we have seen in our studies. So we have had for over 25 years this anti-doping helpline here at the Karolinska Hospital. But right now, unfortunately, we do not have the resources to continue. And to this hotline, both individuals using doping substances, but also next of kin, healthcare people have reached out for help and support. And the general knowledge among clinicians are often very poor and judgmental when it comes to doping substances. So now instead, many go to internet forums for advice. So there is no place for them to seek appropriate care and help. So with an endless amount of money, we could reinforce our anti-doping hotline and together with clinicians and experts, build up a rehabilitation center and medical care unit for these people. And I think there are some good examples here in the Nordic countries. For example, in Norway at the Oslo University Hospital, they have a similar organization. And of course, research activity will be an important task in this new organization to perform studies to learn more about doping in the society.
Absolutely, Lena. I love this big scale vision that you have of what you would do with unlimited resources. And I think you brought up a really good point that doping isn't just a problem in competitive sport, although it is a big one for sure, but it's also a broader public health problem. And I think this is something that a lot of people take for granted or maybe just don't think about in the forefront. Well, Lena, if you have captivated our listeners and they would like to learn more about you and the wonderful work that you're doing, what is the best way for them to do so?
My contact details and a very brief short overview of my research is defined on the Karolinska Institute homepage, so they can just email me. And if they're interested to read more about specific research, I think best way is to look up my publications at PubMed.
Excellent. Well, listeners, definitely check out these resources. Get in touch if you have specific questions. And Lena, thank you so much for joining us today on the show to share your insights and expertise.
Thank you for having me.
It was a pleasure to chat with you, and listeners, wonderful to have you here with us as well. Please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and/or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.